Welcome to our third and final video on translation. So I want to start just real quick with a picture that shows what the central dogma or gene expression is. So remember, we're going to start with a double-stranded molecule of DNA. Only one of those strands is going to be used as a template to create an anti-parallel and complementary strand of nucleotides. We focus on messenger RNA because that's what's used during translation to make our protein. So gene expression, also known as the central dogma of biology, has two main parts: transcription, where we're going from DNA, or we're starting with DNA to make RNA. And then translation is when we're changing languages because we're going from RNA nucleotides, and we're going to be using that information to build proteins, which are made up of amino acids. Yes, we call that translation. So what we're going to do now is look at how I might, or will, ask you to apply this information. And so hopefully this next slide is really going to help you kind of visualize what's happening during gene expression. So I have to be very careful with my mouse so I don't get too far ahead of myself. Okay, so notice here, I am giving you a template strand of DNA. And I want you to give me the messenger RNA sequence and directionality, and the end of gene expression is always a protein. So you're also going to give me the sequence of amino acids that will make that protein. All right, so remember, the template DNA strand is always the 3' to 5' strand. And if I just showed you the 3' TAC, etc., and then the 5', and I said, hey, what is this? You would know that it's DNA for two reasons. The first is it has thymine in it, and only DNA has thymine. And the second is because it runs 3' to 5'. Remember, the messenger RNA is always going to run 5' to 3', and it's going to have uracil in it, okay? So, but in this case, I'm telling you, hey, this is your template DNA. I want you to go through the process of gene expression. All right, so remember, we first need to take this starting template strand of DNA nucleotides and do the process of transcription, where we're going to make a single strand of RNA. All right, so to do this, and I have it kind of up a little bit higher so that the captions wouldn't cover up any of my images. So that's why the RNA is up a little bit higher. So remember, the RNA is going to be anti-parallel and complementary to that template DNA strand. So the RNA is always going to run 5' to 3'. And then now we need to do base pairing. So because the first nucleotide is a thymine, T is going to pair with A. So that's what we have as our first RNA nucleotide. So then keep going. So A pairs with U, C pairs with G, G pairs with C, okay? And then this just continues to happen until we get all the way through the molecule. So this is our messenger RNA. This is our transcript. That is what's made at the end of transcription. So again, pay attention. The messenger RNA is always 5' to 3'. It's got uracils and no thymines, and it's going to be anti-parallel and complementary to this strand of DNA nucleotides. The biggest mistake I see people make is they forget they put the T's here in the RNA sequence, and that is not correct, okay? Make sure you always have uracil instead. All right, so now that we've finished transcription, now we need to do translation. And remember, in order to do that, we need to divide the messenger RNA into codons, which are sets of 3 RNA nucleotides. So remember, the first one is always called the start codon. Then we keep going, and there's codon 2, codon 3, codon 4, 5, and then remember the last codon is always called the stop codon. All right, so now we need to use the codon wheel to figure out which amino acids we need, because remember the sequence of amino acids is really important. So first up is AUG. Now we're always going to have the same amino acid first. It's kind of a way to check yourself on exams. If I'm asking you to do this process by hand, or if it's in the form of like a multiple choice question, your first amino acid is always going to be methionine. But let's practice using this codon wheel. So remember, you always start in the center. So here's the A. My next nucleotide is a U, so going to work our way out. And then the third nucleotide in that codon is G, which is right here. And that's going to give us methionine, okay? So what happens next is we need methionine to be delivered to the ribosome. There it goes. And so remember, methionine gets dropped off at this place because this is my little tRNA, this green cross here, okay? So remember, the transfer RNA has two important sites. It's got the methionine amino acid right here, and then it also has the anticodon. So remember, the anticodon is going to be complementary to the actual codon. So if our codon was AUG, here's our anticodon, UAC. Remember, anticodons are on tRNA. And so this is how methionine gets delivered first, because this anticodon can only go where there's an AUG. So this is the only place in my example where methionine could get dropped off, because it's the only place the anticodon will match. Okay, so now we need to read codon 2 to figure out what our next amino acid should be. So CGU. So again, follow along with the codon wheel. CGU gives us ARG, which is arginine. So that one's going to be delivered second. And notice the anticodon of that tRNA is going to be complementary to our actual codon. So then our next codon is AUU, which is going to be right here. ILE stands for isoleucine. Bloop, there it goes. The other ones were supposed to show that too. That's all right. All right, so now our fourth codon, CUG. So here's CUG is going to be leucine. Here it comes. Again, it can only get dropped off there because its anticodon is going to be complementary to only this codon. And then last but not least, GUA is going to give us valine. Now remember, when the stop codon is what we get to at the ribosome, that means done, stop. There's no tRNAs because there's no amino acid called stop, okay? And so remember, this is when a release factor comes to the ribosome, causing everything to release from one another. And that's when the ribosome disassembles, the chain of amino acids gets released, the tRNA gets released, and everything goes its own merry way. All right, y'all, that is gene expression. Make sure that you understand how to start with a piece of DNA, use it to make RNA, then how you use the messenger RNA, you use that information to go to the codon wheel to find which amino acids you would need. Because the answer to this, what amino acids would be, made, or which ones would they be connected together during gene expression, your answer is MET, ARG, this ILE, leucine, and valine. And again, you'll only have to put the 3 letters, but this is your answer: methionine, arginine, isoleucine, leucine, and valine. All right, y'all, let me know if you have any questions.